Hey guys, so we are looking at chapter 10 today, which is all about cellular respiration. And we're actually going to do this similar to how we did chapter 9. So I'm going to combine here at the start of chapter 10 two sections and do both sections 10.1 and 10.2 with you here first. So that means that of this chapter, this is going to be the longer of the two screencasts. So as we did with chapter 9, if this is something that, of course, you want to start, knock some of these slides out of the way and then come back to the next day, that's, it's posted that way for you on the agenda anyway. And if you were in class doing this with me, we would take several days to go over 10.1 and 10.2. So don't feel like you have to watch this whole screencast again in one day. Because it is going to be fairly long, okay, kind of like we saw with photosynthesis. So go ahead and get the heading here for right now. It's chapter 10, cellular respiration. And if you look at the image here on this first slide, obviously what we'll be doing throughout this chapter is kind of comparing this to what we learned about photosynthesis in chapter 9 and look at, how is cellular respiration similar to that, and also how is it going to be different from what we've been talking about. So one of the things that we've already looked at is organelles and organelle function. That was part of chapter 8. And so as we look at this, obviously we know the chloroplast is where we're going to be doing photosynthesis. Cellular respiration is done in that organelle that we refer to as being the powerhouse of the cell, which we've all learned as the mitochondria. So the mitochondria does cellular respiration, which means that truly any eukaryote can do this process now. We're not just looking at those types of organisms that can produce their own food. And this is any living thing that obtains food from anything, whether it's from its own body or from consuming other organisms, okay? So sections 10.1 and 10.2, your title is Cellular Respiration, and then we're going to move on from that. I'm checking my notes over here to make sure I've got the right information that I'm sharing with you. So here we see a picture of a mitochondrion. It's pretty well magnified. You can see we have some little lines in here that represent the membranes. These are folded membranes on the interior, and we'll talk about in this section exactly what those represent and what they're called, because within this organelle, similar to what we saw with the chloroplast, we're going to have different compartments where different sets of reactions will be occurring. All right, so objectives for these first two sections of chapter 10. We're going to again kind of focus on the big picture item with this, or the big picture items with all of these. So what I mean by that is the individual steps here are not as important as understanding kind of the summary. So in other words, keep in the back of your mind as we talk through all these things and as you're jotting things down today, that what we're concerned most with is, what's the primary purpose of this process and why is it necessary that we use these different steps? What's involved in each step and where does it take place? So I always like to think of the what, the when, the where, the how, and sometimes the why also, right? Because the why is the most meaningful part of all of this, okay? So if we start section 10.1, we're looking at here definition for the term cellular respiration. So it makes sense we want to start with what that actually represents. So cellular respiration is a process of energy conversion. So we're taking food that we've either consumed, or if you're a photoautotroph or a chemoautotroph and you've made your own food, then you would still be doing cellular respiration to break it down into usable forms of energy. All right, and so when we look at usable forms of energy, we're primarily talking about here ATP, that molecule that we first learned about in section 9.1. So we want to make ATP energy using food molecules as kind of our source for creating that ATP from our ADP. The primary choice, right, for making any energy in any cell is going to be to use carbohydrates if possible. And so the one that we tend to focus on the most is the same one that we saw was a product of photosynthesis, which is glucose. And if you remember, that C6H12O6. So this 6-carbon molecule can be used to produce kind of a massive amount of ATP, right? We can get down here 36 ATP through what we call aerobic respiration. And that's in addition to the 2 that we can produce up here through what we call this anaerobic process. And we'll talk about what that means here in a second. But I like this diagram here because it kind of summarizes in one kind of short and simple diagram. At least for me, this looks to be very simple. It has several different stages and labels there, and then I can see real clearly exactly how many ATP I get out of these different stages. Okay. So when we use the words aerobic and anaerobic, these words integrate a couple of vocab terms that we learned about with our Latin roots at the start of the year. If you remember, air right here means air, not such a complicated term in terms of our Latin roots. Air, or you could even think about it in this case as being oxygen. If something's anaerobic, remember a or an means not or without. And so when we look at a process that's anaerobic, that really just translates into meaning something that occurs without oxygen. Okay, so you can see up here, my aerobic stage right here is happening with oxygen present, whereas this one up here in the pink is saying it's anaerobic, which must tell me that it's occurring without oxygen present, as is the one in the green right here. Okay, so make sure you get those two terms, and then we're going to talk through what all of these stages are and what all of these words are that you're seeing here up on this diagram. Okay, so here are those definitions again: aerobic and anaerobic. And when we talk about cellular respiration, obviously, like I mentioned before, we're primarily looking at the mitochondrion. However, we also need to consider that there are some stages that occur outside of this membrane-bound organelle. So it's only actually the aerobic stages that will occur in the mitochondrion. So it's only the oxygen-requiring steps of this process that will occur in this organelle. The anaerobic stages will occur in places that all cells have in common. So when we talk about all cells, what we know they all contain is going to be cytoplasm. And so if it's an anaerobic process, and there are two different steps we're going to talk about that are anaerobic. If it's an anaerobic type of process, it'll occur out in the cytoplasm. So this means that both prokaryotes and eukaryotes have the ability to do this anaerobic type of respiration. When we talk about aerobic, this is specific for eukaryotes only. My handwriting's getting worse there, but eukaryotes only for those aerobic or oxygen requiring processes, because again, those are the only types of cells that actually contain these membrane bound organelles like the mitochondrion. Okay. So within my mitochondrion, I have several different kind of locations and compartments. I have what we call the inner membrane right here, which is kind of this folded wall that we see here. Kind of looks like maybe a wall and something. We have an outer membrane, which is a darker brown color here that surrounds the entire organelle and separates it from the cytoplasm. Cristae is the term that we give to the folds of this inner membrane, and then the matrix is kind of the space between, in the blue here, the inner membrane and the folds there, and then the opposite side. So it kind of sits in between. So in a way, it's like this empty space. It's not fluid filled, but they're representing it here in that kind of bluish color so that it looks different from the membrane folds itself. Okay. So those are going to be terms that you're going to hear me come back to and talk about more as we go through these three stages. And we're going to start with those right here. So when we talk about cellular respiration, we have three steps that occur as part of anaerobic and really what we call aerobic respiration. So when we hear the term cellular respiration, you're generally thinking of all three of these steps, okay? And they do have to occur in a certain order, and they're labeled here in that certain order. So we go 1, 2, 3 for these steps, okay? So you're going to want to get all those. So the first step is called glycolysis. In this diagram here on the right, it's represented by what's happening in this bluish area. And you can see we have some interesting names here. We've got amino acids and fatty acids, which we're familiar with. Then we've got these other things like pyruvate and acetyl-CoA. We have some CO2 coming out. We have some electrons moving. And so obviously there's a lot of different things that are falling into this kind of first step. But we do know that glycolysis in this first step is anaerobic, again, requiring no oxygen. And for that reason, it gets to occur out in the cytoplasm. Okay, so both prokaryotes and eukaryotes can do glycolysis, all right, because every cell type has cytoplasm. Following glycolysis, if there's oxygen present, we can go on into the mitochondria In there, we're going to perform the second step of aerobic respiration, which is called the Krebs cycle. Krebs cycle, because it's aerobic, is going to be requiring oxygen. And really, you can think of any kind of step that's involving the use of oxygen as being aerobic and as also occurring in the mitochondria. So the Krebs cycle occurs in the mitochondria, and specifically, we could say this is in the mitochondrial matrix. Okay, so that's the part of the mitochondria. Remember, it's in between kind of those folds that we see on the inside. The electron transport chain is also aerobic. It also requires oxygen, and that's where we're actually going to produce some water. So this is kind of like the reactants that we use in photosynthesis are what we're actually going to be producing in this process now. So within the mitochondria here, this ETC is not going to occur in the matrix. It's going to occur in the inner mitochondrial membrane. All right, and so that is that kind of folded membrane that got messed up here. It's that folded membrane there in the actual mitochondria, okay, kind of wraps around. That's where we have our ETC. So just like the electron transport chain that we saw in photosynthesis, this has to be occurring in a membrane. It's just that now we're talking about a totally different organelle. So it's the same kind of setup. It's going to be a series of proteins that allows electrons to kind of bounce around and move through it. And then that energy is going to help to power the production of ATP through that ATP synthase that we saw also in photosynthesis. Okay, so this is, again, general overview. Here's your equation for cellular respiration. It's the exact same thing that we saw in photosynthesis. It's just that now we flipped. So the reactants that I'm seeing here on the left-hand side of this equation are the same things that we produced in photosynthesis, all right? And now the products here on the right-hand side of this equation for cellular respiration were actually our reactants that we saw in photosynthesis. So this is very similar to the types of molecules we've been working with, except for now we're not including sunlight. Okay, but beyond that, it's going to be the same types of molecules that are involved here. So when we think about the labels that we're using for this, glycolysis is step number 1, right? And I'm labeling that as step number 1 here. Glycolysis is going to be using, let me go back here. Glycolysis is going to be using glucose, and it doesn't really even produce any of the products that we see here in this equation. That doesn't mean it doesn't produce anything. It's just it's not producing either CO2 or H2O. Okay, so we're going to break down glucose through this process. We're going to create some other things, but we're not actually going to release any products as we see in this equation. Okay. Second step, we're going to use oxygen because it's an aerobic step, and we're going to release CO2 as kind of a byproduct. All right. Now we know that for animals, we tend to breathe these things out like through our lungs. Plants obviously would push out CO2 through their stomata, similar to how they pushed out oxygen. But you know, they could also kind of hold on to that CO2 if they were also actively doing photosynthesis. Water is going to be produced in the last stage, the electron transport chain, or what we call the ETC. And again, because it's also in the mitochondrion, it requires oxygen as well, being that it's aerobic. Okay. So the arrows there, I always think it's helpful to jot this down this way because it's real simple. If I happen to ask you a question on a test, you know, which stage of respiration produces CO2? If you've got this jotted down like this, it's really easy. You can say, hey, I know CO2 is coming strictly from the Krebs cycle, right? And so that would be your answer to a question that looked like that. Okay. All right, so looking at the specific steps, I mentioned that glycolysis is going to be our first stage, and glycolysis is kind of this upper portion of this diagram here. So when we think about what's happening in glycolysis, there is this Latin root right here, LYS, which is the same Latin root we saw in an organelle called the lysosome. If you remember what lys means, it means to split, to loosen, or to break. Oops. And so when we talk about cellular respiration, we're looking at here the breakdown of, and I can't write that, it looks like here. We're looking at the breakdown of of glucose through this process. So let me just start it right here. Okay, so when we say glycolysis, glyco is referring to sugars or like glycogen, carbohydrates, and then lyse or lysis means to break. So we're kind of physically pulling apart our glucose molecule. As we said before, it's anaerobic, so it's going to occur out in the cytoplasm, meaning all cells can do it. And through this process, we're going to create some energy carriers, all right? And so the energy carriers that we're going to try and create are something called NADH, which is very similar to the NADPH we saw in photosynthesis, except here, obviously, we don't have a P. So this is not a typo. This is an entirely different molecule that's lacking the phosphate that we see in NADPH, all right? Same kind of basic structure minus that phosphate part, which is why we're lacking the P here. So we get some NADH that's produced. That NADH is going to play a really important role in that as it's produced kind of here, it's going to go out and around and go down all the way to the electron transport chain. The ATP that we make is kind of already done. It gets added to the grand total of ATP molecules made through cellular respiration. And through this process, we're going to actually burn some ATP to kind of get it started, but then we produce more. So we say we have a net gain of 2 ATP through this process. Okay. So when we look at the individual steps for glycolysis, here is what we've got. And again, as I said with photosynthesis, try not to get too bogged down in the individual steps. Try to focus on, these big picture ideas. So this is kind of the progression. This is how it's going to move. However, keep in mind, what's the purpose? Why are we doing this? Where is it happening? And who's involved, right? Like which molecules are part of this. So one glucose molecule after you consume food, or if you're a plant, after you produce glucose through photosynthesis, it's going to get into the cytoplasm, and there it's going to start to go through glycolysis. So it starts to get broken down, and the initial breakdown, it requires that we invest 2 ATP right here. So in this case, we've kind of just used 2 ATP. We've not actually created anything. We've just gotten rid of those 2 ATP, which this means that so far we are like -2 on our number of ATP produced through this process. We've actually lost ATP. Now, where that becomes beneficial is down here, where I'm getting my energy carriers and my ATPs that are going to be produced. So even though I've burned off 2 ATPs through several steps, and there's more here than what you're seeing, I end up producing actually 4 ATP, and so my net gain as a result of that is going to be +2. So I actually create 2 total ATP through just the breakdown of glucose, which is not a lot. But as we move through the rest of this process, you'll see that our numbers will increase like significantly, okay? The NADH, like I said, is an energy carrier. And so this energy carrier is going to go on to not the next stage, but all the way down to the electron transport chain, to stage number 3. Okay, and there it's going to be used to donate energy to those electrons that are bouncing through that chain to help create ATP. Okay, so we rearrange carbons basically. We make a net gain of 2 ATP, and then we also make 2 NADH. Now, as this happens, glycolysis, remember, is out in the cytoplasm, and one of the products of glycolysis is this thing called pyruvate. So pyruvate is like a 3-carbon molecule. It's right here. Pyruvate is kind of unique in that it doesn't have the ability to get into the mitochondria without a little bit of help. And so the way that it can enter the mitochondria is to be converted into this other thing called acetyl-CoA. When that happens, we actually release some CO2 as a byproduct that gets breathed out, and then we create an additional NADH as an energy carrier, which again will go on to the ETC. Okay, so there's a benefit to having to do this in that we also, can produce these 2 products, but it is really necessary that we add this little tag here. This is my acetyl-CoA that then can enter the mitochondria. And we're about to see that here in this image. This is going to kind of continue in this direction. Okay. So this, again, out in the cytoplasm is still anaerobic, but here now we're pulling off a carbon. We're adding a little tag and modifying the molecule to make something called acetyl-CoA. And again, through that process, we make 1 additional energy carrier. Okay. Second stage of cellular respiration is called the Krebs cycle. The Krebs cycle also kind of goes by some different names, but you will almost always see it called the Krebs cycle. This is going to be in the mitochondria again, but here this time it's in the mitochondrial matrix, and I mentioned that before. So that is kind of like here in the little folds of this membrane. In that mitochondrial matrix, and kind of, sorry, I should say between those little folds of the membrane, we have to have oxygen available, okay, to run this process. We have to have the acetyl-CoA that came from glucose, which would be, kind of this part right here. We have to have really some kind of starter molecules that start out in that cycle that are going to continue to kind of cycle around. Through that process, we're going to produce a couple more ATP, and let me move myself around here a little bit. We produce a total of 2 to 4 additional ATP molecules. So that means we've got 2 ATP here. We're going to get 2 to 4 here. And beyond that, we're not sure. We're going to have to look at what we get out of the electron transport chain. But remember, a while back, I said we weren't, we were going to produce a significant amount, much more than just 2, okay? So looking at our steps of the Krebs cycle, we have several different steps, and similar to what we saw in the light-independent reactions, right? This is a cycle. There's a lot of carbons that are going to be rearranged here, and I'm not giving you a lot of names for those because I'm not interested in you trying to kind of memorize each individual chemical reaction and enzyme and everything else that's part of this process. It is super complicated. But for your purposes, kind of the general ideas that you want to keep in mind is that we're using our product from glycolysis. So this is my connection here to glycolysis. My acetyl-CoA comes in. I'm going to use some oxygen, and I'm going to start rearranging some atoms here, and I'm going to make more energy carriers. So I get, again, ATP. I get more NADH, and then I get this extra thing, which I've not seen before. This is called FADH2. So this is going to be another example of an energy carrier. If you remember what we did in chapter 9 when we talked about energy carriers between the light-dependent, the first stage, and the light-independent, the second stage, I had 2 energy carriers. We had NADPH and we had ATP. Here we also just have 2. We have NADH and FADH2. ATP itself in this type of series of reactions is not an energy carrier. It is like the end goal. It is a whole reason why we do cellular respiration, okay? So it doesn't serve as an energy carrier. Only my NADH and my FADH2 can serve that role, okay? So those are going to be produced here, and then just like we saw with glycolysis, they move on to the third and final stage, which is the ETC, okay? So if we look at the Krebs cycle as kind of an overview, this part over here is what was happening out in the cytoplasm, right? This was kind of our follow-up from glycolysis, where we had to add that CoA tag to make acetyl-CoA that then could go into my my Krebs cycle. So this right here is my Krebs cycle, right? I'm kind of revolving and moving around this. So you can see all of the things that are going to be produced. I'm getting an NADH here. I'm getting a CO2 here that gets breathed out, right? I'm going to get more NADH here. I'm going to get an ATP, NADH and FADH2. And so remember, all of these together go on to the ETC. Okay, ATP is just done as it's produced. So you get lots of different things kind of coming out of the cycle, and certainly, again, because it's a cycle, it's just going to be kind of this continuous process that will continue to occur as long as we have the ability to do it, okay? Last stage is called the electron transport chain, also known as the ETC. So instead of saying electron transport chain, a lot of times we just say ETC. The ETC is similar to what we saw in photosynthesis in that we're dealing with a number of proteins in this inner mitochondrial membrane this time. So unlike in photosynthesis, where this was in the thylakoid membranes that make up the grana, this is in the inner mitochondrial membrane. And this is, again, a series of proteins. And those proteins have one main job, which is to allow energized electrons to move through them to the point to where we can create a concentration gradient that can then drive the production of ATP with the help of ATP synthase. So the ETC, unlike the other types of stages that we talked about, can create a ton of ATP. So we mentioned earlier, just to review, you get 2 ATP from glycolysis, you can get 2 to 4 ATP from Krebs, and now I'm going to say you can get 32 to 34 ATP from this last stage. So if I were to ask you, what's the most, which stage of cellular respiration produces the largest amount of ATP, your answer would have to be the ETC, because it's not even comparable to 2 or to 2 to 4. So 32 to 34 is significant, and this is really the reason why we do glycolysis in Krebs, is to create the energy carriers that we need to be able to do this set of processes. So I've got 4 steps here for this one. Again, don't stress about the steps, but just have an idea of the general kind of process. So we use our energy carriers right here, my NADH and my FADH2, as energy carriers. Their role here is going to be to donate electrons to the proteins that sit in the inner mitochondrial membrane as part of the ETC. Those energized electrons move through the membrane and the proteins there, and that allows for the active transport of hydrogen ions again, or the pumping of hydrogen ions across the membrane. At the end of the ETC, just like we saw in photosynthesis, electrons have to go somewhere, and this time they're going to go to our oxygen, and that will then create water as a byproduct. So oxygen, remember, this is aerobic. This is where that reactant comes into play, and this is where we produce water as a product of this process. To get ATP, same as what we saw before, hydrogen ions diffuse down their concentration gradient using facilitated diffusion and through a channel protein called ATP synthase. And as that occurs, the spinning of that ATP synthase protein allows ADP to be made into ATP. Right? So looking at the kind of general overview of these steps, here is kind of what this looks like. And this should maybe look familiar to you, especially if you remember seeing the one for sections 9.2 and 9.3. It's just now I'm not using green to represent these colors. I'm using this brownish, reddish kind of color, okay? So I've got NADH and FADH2 here. They're donating electrons to the ETC, and those will kind of bump through. And as they reach the end of this ETC, those 2 electrons have to go somewhere. They can't just float and be by themselves. Where they end up going is to this half of an oxygen, to one single oxygen atom. And with the help of hydrogen ions, we're going to create water, which then gets released, right? It's just kind of like a waste product that can be released as a result. As we saw with photosynthesis, the pumping of these hydrogen ions across this membrane results in a concentration gradient. So here I have a high concentration of hydrogen ions, and here on this side I have a low concentration of hydrogen ions. And so when they diffuse from high to low, this is where we get the creation of ATP. And remember, it's a significant amount of ATP. It's 30 to 32, or you could even say 32 to 40. All right, it's going to be a large number of ATP molecules. So this should look very similar to what we saw in 9.2, 9.3. If it doesn't, it would be worth your time to go back and also look at it there, because you may see this appearing on your test in a number of different formats, because your test is over both 9 and 10 together. All right, so here's kind of my measure of how much ATP I can make from a single glucose. So glycolysis makes 2. Krebs cycle will make 2 to 4. The electron transport chain makes 30 to 32 ATP. It also releases some water, and we get some CO2 out of Krebs. So in total, if we think about this as a range, we could produce anywhere between 36 and 40 ATP molecules per glucose. And that's a significant number, right? 36 is a nice significant number. And now we're even saying we can go above and beyond that. So how would you get to 40 versus 36? That has everything to do with how much oxygen you have available in your cells. So if you're exercising harder, and you're kind of deprived of oxygen, maybe, right? You're breathing heavily to try and move oxygen through your blood vessels to all of your cells. You may run low on some oxygen and you may make, 36 or maybe below 36. But if you have, the correct amount of oxygen, the proper saturation of that, you can make up to 40 ATP. All right. Either way, if it's 36, 37, 38, or 40, I mean, we're looking at a significant number regardless. All right. So this is my last slide for sections 10.2 and 10.3. If you've got any questions, as usual, don't hesitate to let me know. And I guess that's it for right now. So I will see you all later. Bye.